My name is Jon Foss-Skiftesvik, and I'm a neurosurgical resident at the Rigshospitalet University Hospital, and also now a PhD student looking at pediatric central nervous system tumors in collaboration with the State and Serum Institute. I'm going to tell you a bit about some of the results from a recent genome-wide association study we did on children with brain tumors, brain and spinal cord tumors. So pediatric central nervous system tumors, they constitute the second most common form of malignancy during childhood, second to hematological cancers. But they cause the most cancer-related deaths in the pediatric population. Genetic predisposition has long been suspected as an underlying cause. We do not know much about the underlying causes of these diseases. And over the recent couple of years, we've gained experience in terms of rare high-risk mutations, because next generation sequencing studies have shown that 8 to 10 to 12% of children with cancer harbor an underlying germline mutation in a cancer-related gene. But risk attributable to common variants have received less attention. And the best way of assessing such risk is by conducting genome-wide association studies, which we've heard some of the other researchers here talk about today and the previous days. And these are studies where you assess or you screen the genomes of a large number of well-defined cases and compare them with controls. And you basically look at thousands, hundreds of thousands of single nucleotide variants to see if they differ in their frequency between cases and controls. They might be associated with this disease in question, but not necessarily causative. Such studies, GWAS studies, have been conducted for some of the CNS tumor types in adults, but results from these studies are not believed to be transferable, as the pediatric CNS tumors differ vastly in molecular biology from those of adults. So what we wanted to do was to investigate the potential for shared genetic susceptibility across pediatric CNS tumor types attributable to common variants. We also wanted to look whether or not this risk differentiated between different age of onset groups, but I won't go too much into that in sake of time. So what we did was a nationwide GWAS, including cases that were diagnosed for more than over 30 years. We could only do this by combining the Danish Childhood Cancer Registry with the Danish National Screening Biobank, which is a part of the National Biobank at State and Serum Institute. So more specifically, and I don't want you to read all this, but more specifically, we pulled the personal identification numbers of all children diagnosed with a CNS tumor in Denmark before turning 15 years of age, years 1985 through 2017. Then we excluded those who did not have an existing and available dried blood spot sample in the PKU Biobank at State and Serum Institute. We also focused at the most relevant diagnosis. So we ended up with a case cohort of approximately 1,100 cases. What we then did was we genotyped the dried blood spot samples of these cases using the Illumina GSA array, which contains approximately 1 million single nucleotide variants. And for controls, we have randomly selected more than 4,000 controls from a population cohort in the ISAC sample, which we heard about, at least for those of you in the PhD course, we heard about that yesterday. And in these cases, those controls had undergone genotyping using the same array previously. So when looking at results from genome-wide association studies, you're almost always presented with a Manhattan plot. And we've seen a couple of these during this morning, but for those of you who are not really familiar with it, on the x-axis, you have the chromosomes. On the y-axis, you have the negative log to the p-value. Each dot represents a single nucleotide variant. So as you can see here, most of these do not differ markedly from cases to controls. They're common variants. But some places you see high rises such as this, the so-called high rise, which really is a lot of SNPs that differ in frequency between cases and controls to a higher degree than the others. And all of these SNPs here, they map to the same part of the genome in chromosome 1 to a gene called PAPA2. I'll get back to that. So if we look at, this was the overall cohort, all cases. If we focus on the ones in the late onset group, defined as children being diagnosed between ages 4 and 15, we see the same signal, it just intensifies in strength. So the p-value here for this PAPA2 association was 9.7 times 10 to the minus 8. So that is close to, but not below, the genome-wide significance threshold. So the commonly accepted genome-wide significance threshold is 5 times 10 to the minus 8. It is under debate and being accused of being overly conservative, as it somehow assumes that all of these SNPs are independent analysis, or are independent. Not all of them are, because some of them are inherited in linkage, but I won't discuss that further. I just wanted to say that what we did was really implicate this locus and another one I won't talk too much about as potential susceptibility loci for CNS tumors in children. And even though they didn't retain the genome-wide or the significance following correction for multiple analysis, we believe the results to be strong enough to warrant further investigation. So if we look at the PAPA2 gene specifically, this is a protein coding gene on chromosome 1. It is expressed in brain tissue. And what it does is really, and you can rest assured, I'm a better neurosurgeon than a graphical designer. But what it really does is it cleaves IGF-1, insulin-like growth factor 1, from its binding proteins 3 and 5, which results in free IGF-1, which is able to bind to its receptor. So alterations in PAPA2 activity could potentially change the local concentrations of IGF-1. And PAPA2 has not been linked to cancer before, but IGF-1 has been shown to be associated with a wide range of pediatric cancers, including brain tumors. We see increased levels of IGF-1 in tumor tissue from astrocytoma, a brain tumor, which is correlated with the degree of malignancy of the tumor. We also see the binding proteins 3 and 5. They're increased in cerebrospinal fluid of children with certain forms of brain tumors, such as ependymoma and medulloblastoma. And on a side note, PAPA2 is also associated through a large GWAS study with body height, and body height is itself associated with the risk of glioma, which is one of the most common form of brain tumors. So briefly addressing the strengths and weaknesses of this study, we have the largest published GWAS cohort for pediatric brain tumors. We sampled cases for 30 years, or included cases diagnosed for more than 30 years nationwide. And we're pretty happy with the population-based controls. They're unbiased toward health, but also receipt of social benefits, social class, etc. Many other international studies use control cohorts from other disease populations, such as they have a diabetes cohort they can use as controls for cancer cohort. We were lucky enough to get around that. I'm jumping in the sake of time, but we'll look at some of the weaknesses. Still, the sample size, even though it was large, it wasn't big enough to achieve genome-wide significance. And I guess it's a good thing that not more children suffer from these horrible diseases, but still it impedes research in this field. There has also been a number of alterations, updates of the classification systems used to classify CNS tumors over the last 30 years. But as we approach this with a pan-tumor approach, we do not believe that this has had significance to our results, as most of the changes have been within different tumor subtype categories. I'll leave some of these for the potential questions afterwards in the sake of time. Concluding remarks here, we have conducted, and this was just accepted for publication, the first population-based genome-wide significance, genome-wide association study for children with central nervous system tumors. And we've implicated 2 loci to be involved, two loci which have functions in CNS development and in the IGF axis. And these results really can be viewed as the discovery cohort of a GWAS. They need to be validated in larger data sets. And we've used all our cases. So that's really an issue that is coming back to haunt us all the time with these studies on rare diseases. We hope by publishing this that we will initiate some more collaborative research internationally, so we can get these findings validated and to build bigger cohorts. Finally, I want to thank my collaborators at Rigshospitalet and the State and Serum Institute, and all of you for listening.